today our chapter part 2 of sexual reproduction in flowering plant. So today we will start with pollination. So what is pollination? Transfer of pollen grains from anther to stigma, right? Means pollen grain from male part goes to female part stigma. So this is called pollination. There are two types of pollination, self pollination and cross pollination. What is self pollination? Self means same flower of the same plant, right? Like we have seen hibiscus, China rose, it is a bisexual flower, male part and female part both are present in same flower. So it is bisexual flower. So in cell pollination, flower should be same of the same plant. Again, cell pollination are divided into two, autogamy and geitonogamy. What is autogamy? This is very important topic, right? So pollination means transfer of pollen grain from anther to stigma. Two types, self pollination and cross pollination. Self pollination, flower, same flower or different flower of the, but plant should be same. Like this is the plant, this is the flower, right? One more flower is here. So from this here to come to this. So this is also we can say self pollination because plant is same, right? So cell pollination and cross pollination. Cell pollination, same flower of the same plant. Cross pollination, two flowers of different plants of the same species. Means plant is different but species is same, like hibiscus. Again, cell pollination, autogamy and geitonogamy. What is autogamy? Means pollination in the same flower. Flowers should be same. So this is the, suppose this is the flower, right? And this is the female part, stigma, style and ovary. Let me clear this. This is the flower, right? And this is the female part and this is the male part. So in one flower, if transfer of pollen grain to going to stigma part of the pistil, then it is autogamy. What about geitonogamy? Plant should be same but flower is different, right? Flower is different. So autogamy, pollination in the same flower. Again, in autogamy, there are two types of flower. Like in viola, example, oxalis, commelia, they produce two types of flowers, which is called chasmoglamous and clistogamous. What is chasmogamous and what is clistogamous? Diagram is given in the NCERT. Chasmogamous flower is opened. Chasmo, M, masculine, just to learn. Chasmo, M, masculine, they are always open. Clisto, like female, which does not open at all, right? So in Gasmogramus, flowers are open. For this, anthers and stigma both are right exposed in Chasmo, but they are not exposed in Clistogamous. So in chasmogamy, synchrony in pollen release and stigma should take place and arrangement should be close. And in clistogamy, there is no chance of cross pollination takes place. Why? Because clistogamous flower does not open. If it is does not open, so there is no chance of cross pollination to take place in clistogamous flower. So the viola, oxalis, commelia, two types of flowers are there, chasmo and clistogamous. Now cross pollination, in cross pollination, two flower of different plant of the same species, means plant is different. One plant is here and another plant is here, suppose, right? So there is transfer of pollen grain from one plant to other plant of the same species is called cross pollination, and xenogamy is also it is a type of cross pollination. Next, so is it clear? So one more thing, pollination, because transfer of pollen grains from anther and stigma is taking place. Gametes are non-motile, so obviously some pollinating agents required in that. है ना? Pollinating agents will be required. So next topic, devices for autogamy means how pollination takes place in the same flower. There are some conditions for this, and there are some conditions for xenogamy also. So for autogamy means for cell pollination, what are the requirements? Simultaneous maturation of anther and pistil should be there. Pistil and anther, मैच्योरेशन साइमल्टेनियसली, ऐसा नहीं कि ये जल्दी हो जाए या ये, तो इट विल नॉट, सेल्फ पॉलिनेशन कैन नॉट बी टेकन प्लेस, इट विल बी टेक प्लेस while both are mature at same time. Anther and stigma should close lie to each other. For self pollination, the orientation should be very closed, right? And clistogamous flower, oxalis commedia viola, there is always autogamy will take place. Why? Because clistogamous flower is, which flower? It does not open, it is a closed flower. In closed flower there is no chance from anther to come from outside, so it autogamy always takes place in clistogamous flower. So for autogamy, what are the condition? Lie close to each other, clistogamous flower, and maturation of same anther and pistil maturation at same time take place. Now for xenogamy means we are talking about cross pollination. So unisexual flower, obviously flower should be unisexual, na? Only one male part or either female part is there, so it will go to other part or other plant only. So for xenogamy or for cell cross pollination, unisexual flower and dioecious plant required. Pollen release and stigma are not synchronized, synchronization is not needed, but orientation or different orientation of anther and stigma of flower should be there. And self incompatible, if it is not incompatible, then also it will go to xenogamy. Now we are talking about pollinating agent. These are the very important question which will be frequently asked in practical also and in exams also. Pollination is one of the most important, which I have already started there at the starting. So wind pollinated flowers, and how we talk about wind pollinated flowers. See, pollen grains, if it is carried by wind, it should be non-sticky and it will be very lightweight also. Agar if it is heavy weight, it will not carried away by wind, right? So what are the properties of wind pollinated flowers? Pollen grains should be light. Why it is should be light? Because so that by the wind it can be carried very easily, and it should be non-sticky, right? Dry, sometimes winged also. Well exposed anther, if exposed anther will be well exposed, then also it will be very easily carried away by wind. Stigma should be large feathery, flowers arranged as inflorescence and single ovules. So these are the some. So wind is also, it is a pollinating, it is helping pollination. It is pollinating agent. There are two types of agent, biotic and abiotic. I am starting with abiotic agent, where water and wind is required. And in biotic, birds, insects. So these are the pollinating agent, birds, insect, which are biotic agent, right? So example of wind pollinated is also maize, is an example, maize. Next we will talk about water pollinated flowers. Obviously in water it will be mucus is there, so that it will not be wetted, है ना? Pollen should not be wet, because in water pollinated flowers, examples in wind pollinated example is maize, but here water pollinated example is Vallisneria and Hydrilla zostera, right? And the pollen grain should be it should be mucilaginous coating. Why? So that pollen grains should not be wet, because it is water pollinated. So what will happen in Vallisneria? These are the example. You have to learn this water pollinated flowers. In wind pollinated, non-sticky, lightweight is, and exposed anther, winged, है ना? And in water pollinated, it has to be mucilaginous coated, so that it cannot be wetted. In Vallisneria, male flower released on water surface. Suppose this is the water surface, right? It is the water surface. So male flowers released on water surface, and female flower reach the surface with long stalk for pollination. Same in seagrasses, pollen grains are long ribbon-like and carried passively to submerged female flowers, and then fertilization takes place in that. Next, so this is the now adaptation in insect pollinated flower, which is a biotic pollinating agent. For insect pollinating agent, what will it requires? Fragrance, color. So it should be large, it should be brightly colored and showy. If flowers are small, and small grouped into inflorescence, it should be highly fragrant. If fragrance is there, then only insect will come, right? And it should produce nectar. And yes, one more thing in insect pollinated flower is, in wind pollinated it should be non sticky, but yes, in insect pollinated flowers it should be sticky, so that it can be stick on insect body, right? So it produce nectar, sticky pollen should be there, provide rewards to animal pollinators such as nectar, food, or provide safe place for laying eggs, like in yucca and yucca plant and moth. I will tell you here also. So how insect pollinated, both are benefiting from each other. So what animals are getting? Nectar, right? And what flower is getting? Pollination. So in laying eggs, moth and yucca, what will happen to your yucca plant and moth? In yucca and moth, yucca plant provides moth lay eggs in yucca plant, and it takes, and what yucca does, moth help in pollination. So this is clear. Provide pollinator isn't such as nectar, food to animal pollinator, and safe place for laying eggs. They moth can lay eggs in yucca ovule part, locule part, and yucca. What yucca will do? Yucca is and moth is helping for self pollination.
A few moments
Okay.
later.
So how now pollen pistil interaction? How pollen pistil interaction takes place? In pollen pistil interaction, who decide? Actually, one pollen and pistil should be compatible. Uh, right type of pollen lands on pistil. Okay, that is compatible, and if wrong types of pollen is coming, that is incompatible. Now, who will decide that it is compatible or not? Pistil decide that it is compatible or not. So recognition of compatible pollen is done by, this part is done by pistil. So how it does? By certain secretion of chemicals, pollen pistil interaction takes place, right? Now pollen, how it is going, so see here, suppose this is the part, pollen grains are coming and landing on this stigma part. Pollen grain is getting nutrition from the stigma chemicals, and then pollen grain, what pollen grain is doing? Pollen is germinating and forming pollen tube. Now pollen grains land on stigma, forming pollen tube, and from this tile part it goes to this region, right? And where this region is, suppose this is the egg part, synergid part, and synergid part is there in micropylar end. So pollen tube have two male nucleus. First see the graph, then we will discuss it later on. Two male gametes are there in male gamete, two in pollen tube, one male gamete, and filiform apparatus is guiding them, like in hotel waiter and everything they are guiding na. So this pollen tube filiform apparatus is guiding them, and then pollen tube is coming, and one male gamete will form here, fuse here, and one male gamete will fuse with polar nuclei. So one male nucleus is when it is forming and fertilizing with the egg. This process is called syngamy, and when one male gamete is fertilizing here, so two polar nucleus are there, we have seen, like N or N, and one male nucleus also coming then, so it becomes 3N, means triple fusion. So two types of fusion are taking place, triple fusion and syngamy. So these two types of fertilization, therefore it is called double fertilization. This is called double fertilization. How it is taking place? Pollen grains are germinating, forming into pollen tube. Pollen tube have two nucleus, and one is forming here. One male nucleus going there, and one is going there. So it is forming syngamy, which will develop zygote, and this is developing triple fusion, and both are called double fertilization. In pollen moves into the tube, two male gametes are there in pollen tube. Pollen tube grows through the stigma and style by secreting enzymes and enter ovule through micropylar end. I have told you in diagram that it goes to micropylar end. It enters the embryo sac through filiform apparatus. Filiform apparatus is guiding of synergies to liberate male gametes. What pollen grains germinated? Vegetating nucleus, two male gamete, which is fully developed female gametophyte. And here fertilization, double fertilization take place. Now what is double fertilization? Release of two male gametes from pollen tube into cytoplasm of synergid. Fusion of one male gamete with the egg cell is called syngamy, and which is forming zygote 2n. Male n, female egg n and n plus it forms 2n. See, and which develop into embryo. So zygote, after zygote, what will form? Embryo, right? So zygote is 2n. On how it is coming? For one male gamete going to synergid region, two synergids are there and one egg cell. So it is forming in this egg cell. Second male gametes with the middle part polar nuclei and n plus male gamete n. It forms 3n, which is called primary endosperm nucleus. So syngamy triple fusion occurs in an embryo sac, therefore it is called double fertilization. And central cell is called primary endosperm cell, which develop into endosperm. So what is this endosperm? Actually, this endosperm is having a lot of reserve nutritive material food, which will give nutrition to this embryo, right? Which will give this nutrition to embryo. So what happens? First endosperm take place, after that embryo will start forming. So this is the double fertilization. Post fertilization, what will we will study in first part: pre-fertilization, fertilization. Now post fertilization will take place. Development or endosperm, which is 3n, how it forms, you have already studied. Embryo development will take place after zygote. Zygote se kya bana? Embryo. Maturation of ovule into seed. Seed will forming. What seed is forming? How seed is becoming? Ovule is giving seed and ovary to fruit. Ovary is converting into fruit. So ovule into seed, ovule into form this, and development of embryo and development. There are we will study fruit also. True fruit are there, false fruit are there. True fruit which is developed from fertilization, right? And false fruit which develop from like thalamus, etc. So endosperm. Embryo development precedes embryo, means endosperm development formation takes place early than embryo development. There are three methods of embryo development: this nuclear, cellular, and halovial. What will happen in nuclear type of primary endosperm nucleus? First divide mitotically, cytokinesis, then first free nuclear division takes place, and then cellular. What will happen in that coconut? Coconut water is nuclear endosperm and the white kernel part is cellular endosperm, right? And it is, coconut is very much nutrition, and we are using coconut in food, oil also, and many other, in creams, in hair oil. So because it is very nutritive. And so like this nuclear type primary endosperm nucleus divide mitotically without cytokinesis. First it is, therefore it is free nuclear. After that cell wall formation takes place and endosperm become cellular. So nuclear type example is coconut water, and that white part, kernel part, is called cellular endosperm. And what endosperm is doing, it provides food to developing embryo. One more thing, there are two type of endosperm, albuminous and non-albuminous. If endosperm is completely utilized by embryo, if embryo taken all the endosperm completely, there is no left endosperm, then it is called non-albuminous. If it is present, right, if seed is non-albuminous, if it is present means left, then seed is called albuminous. Okay. So these is the endosperm formation, how it is nuclear division. See here, water, and this is exocarp, mesocarp, endocarp in coconut. Primary endosperm, successive nuclear division, free nuclear endosperm. Then after that cell wall formation takes place. And in cellular, first division followed by cell wall and halobial, transverse wall, micropilar and chalazial chamber, it takes place, right? So this is the endosperm formation. Now embryo development, I have told you, first endosperm development take place. After that, after endosperm, embryo development take place. So from ovule embryo is developed. See, this is the part, we will see, this is the globular part, then it will convert to heart, then torpedo, then it will form mature seed. Okay, so embryo development take place like this. Embryo formation start after endosperm formation. Zygote first divide mitotically and form proembryo. Then it develops, zygote divides, and then it develops into globular, then heart shape, then torpedo, and then cotyledon. See here. Then horseshoe shaped mature embryo with one or two cotyledon. If one cotyledon is present, monocot. If two cotyledon is present, then it is dicot. Now, one apomixis, or we also say agamospore. What are these? When seeds are formed without fertilization. Without fertilization, if seeds are formed, right? Seeds are formed. How seeds are formed? With, we have already studied with ovule. See here. Development of embryo, maturation of ovule into seed, and fruit develops from ovary, which is the part of post fertilization, right? So seeds are formed without fertilization. If there is no fertilization, still seeds are formed. So this is apomixis. It may develop if a diploid egg cell develop into embryo without fertilization. And how it formed? Why without fertilization? When nucellus, you remember nucellus, integuments, inner part, and reserve food material is also there in nucellus, and nucellus, inside nucellus embryo sac is present, right? So if cells of nucellus may develop into embryo and pushed into the embryo, then it is called apomixis and agamospore, right? Now special polyembryony. What is polyembryony? If more than one egg may form in embryo set, then poly means बहुत सारे, poly means many, embryono means embryo. So poly means many embryo. So if more than one egg may form in embryo sac, if more than one embryo sac formed in an embryo, other cells like synergid or nucellus develop into embryo. So this example is orange, lemon, mango, onion, groundnut, etc. So this is the chapter we have finished. I hope you like the video, and thank you. Now we will meet for next chapter.